All right, we're going to talk about patterns of inheritance today, and we'll briefly switch to organismal biology, where we'll talk about whole organisms because we have to talk about patterns of inheritance. But we'll get back to talking about cell and molecular biology. But it's to connect inheritance with cell and molecular biology, I'm going to tell you a bit about the history of studying inheritance. And that plant that you, or the picture you see there, is a garden pea. And this was used to understand the inheritance. And you've probably heard of Mendel before, but maybe maybe you haven't heard of all the difficulties with understanding inheritance and kind of the connection, the difficulty in making connection with what was going on. So we're going to talk about Mendel's laws of inheritance. We'll talk about alleles and some simple genetics things. Um, and this is closely related to sexual reproduction and meiosis because, um, we and sexual reproduction was known of course, at the time that they were trying to figure this out. It was known that had a contribution from both the male and the female parent, but there were problems with inheritance because new individuals resembled the parents in many general ways, but sometimes they didn't. It was hard to predict. Sometimes new traits would appear and then disappear, and so it was hard to tell exactly how things were inherited. And I mean, among the earliest things that they discovered when they looked at microscopes were cells, and they looked at sperm and egg cells and pollen. And so these were well known. It just wasn't known how inheritance worked. Um, and so remember, meiosis was a cell division that reduced the number of chromosomes, and that was known. But the shuffling of chromosomes and the shuffling of genes on chromosomes, that was all unknown. Um, and so it took Mendel to figure this out. And it's kind of amazing because his work went undiscovered for almost 50 years before someone looked at it and said, oh my gosh, this explains everything. And so, in some ways, he kind of seems like a genius, but really, he's not. But it's helpful to think about when there's big advances. Is the guy a genius, or how does the advance happen? And so, there I've got a picture of Elon Musk. He's the owner of Tesla, the auto brand that's making electric cars, really good electric cars. He's also the owner of Solar City, a big solar company that's installing solar powered, um, solar panels on the roof of houses and buildings in the western part of the United States. And he's in charge of SpaceX, the company that's really replacing NASA, building big rockets and making big advances in this. And sometimes he seems like a genius, like this one guy. But if you look at it, he's really not. I mean, um, he's actually kind of, in some ways, kind of a screw up. But he made a lot of money in the internet. Um, he's the one of the founding guys of PayPal, and so he's used that money to invest in these high tech companies. And with you know some progress and some backsliding, he's generally been pretty successful. And so if you go to that link there on the PowerPoint, the SpaceX link, it'll show one of his rockets doing a touchdown on a barge, which is pretty amazing. And then I think there's plans to go to Mars. And he seems like a genius, like holy cow. But then you look at Jeff Bezos, who got all his money from Amazon.com. He's also doing similar things. He's got a company called Blue Origin that's doing the exact same stuff. They're giving SpaceX a run for their money, and they're both far ahead of where NASA is. So maybe they're not geniuses. Maybe they're just in the right place at the right time, and they're heading up efforts that are by lots of people, and so they're able to make this kind of progress. Well, that's kind of the case with Mendel. Inheritance seemed too complex to understand, with the traits disappearing and appearing, um, and the dominant scientific theory at the time was um, that there were fluids. Inheritance was carried in a fluid, probably semen, and there was a blending between the male fluids and the female fluids. But there were some problems with those ideas because you had traits that seemed to disappear and then reappear whole. And really, what Mendel's advantage was he was a farmer, and he was an agriculturist, and agriculture was really advancing in the 17 and 1800s. They were working on the same problem in a very practical way. They were breeding plants and animals, keeping track of how fast they grew, what type of features were there. This was selective breeding, and they were producing new breeds of cows, new breeds of horses, new breeds of dogs. They were improving crop breeds. And so they had an idea of how to do this and how inheritance worked. They didn't have a theory of it, but they had some ideas of how it worked. And Mendel's advances really came right out of agriculture. He selected plants he was very, very familiar with, garden peas. He followed very specific traits like flower color, seed coat, seed color, plant size. And he knew these traits really well. And like farmers were doing, he was keeping track. He was counting. He was looking at the exact numbers of seeds were produced. He'd grow them all up to see what the plants were. And so he was really familiar with his study organisms. And it wasn't luck. He knew how to fertilize the plants. He knew how to control the fertilization. And because of these things, and because he could keep track of these numbers, he then used the mathematics to make some inferences about what was happening. And so he developed two great laws of inheritance, that inheritance was particulate, and that traits segregated independently and assorted independently. And so he made some really huge advances. It seems like it comes out of nowhere, but it comes out of agriculture because he knew his subject. And his approach was an approach that was used in agriculture. He called it a test cross, but it was really a form of backbreeding. So if a trait would disappear, if you found a really desirable trait in an animal and it disappeared in the offspring, you would assume the trait was still there but hidden. And so you would take that offspring and breed it to a relative. And so you would control the breeding. And this is an experimental approach. And what Mendel did was he got true breeding parent plants where he knew the traits, he had followed them for several generations, and he was very confident that they only carried the traits, the inherited factors for these specific traits. And then he would cross them with a parent plant that had a different trait, and he would count the offspring. And so he produced a mathematical offspring. And so the pattern you see here is very typical. He's looking at flower color, and he takes a true breeding purple parent and he crosses it with a true breeding white parent. And he could do this because he knew the flower shape. He could pull out the flowers and fertilize them and then seal them. And he found when he did this that usually all the offspring had one trait, and that one trait, the other trait disappeared. He referred to the trait that showed up as dominant and the one that disappeared as recessive. And he wouldn't stop there then. He would allow the offspring to self-fertilize. Okay? And he found then that the recessive trait, the trait that disappeared, would show up again. But it would show up in a very particular way, that 3/4 of the offspring had the dominant trait and 1/4 had the recessive trait again. And then he would allow these plants to self-fertilize, and he found that some of the purple plants were true breeding. All their offspring were purple. Some of the purple plants again had the 3-to-1 ratio of offspring, three dominant traits to the recessive trait, and that the recessive trait then was pure breeding, or a pure breeding trait. So this test cross, this experimental approach, he determined that the inherited factors, which he called alleles, we would now call them genes, these were discrete. They didn't blend at all, that they were inherited whole and intact, and there was no blending. This was a really important insight because it meant that they were there, they were not lost, and that they came in two types: dominant, which would be always expressed if they were present, and recessive, which could be hidden by dominant. And he made a really important distinction that the physical appearance of the plant could be different from what inherited factors it held. And so we call this now phenotype. The physical type could be different from the inherited factors, the genotype. And so this was really important. These were important ideas, and it explained a lot. It explained traits that appeared to skip generations or would go missing for a long time. Okay, so he did this thing mostly with monohybrid crosses, where he studied two variations of a single trait. And he did this for 7 different traits, each one having just 2 variants. Okay, so he did this for flower color, seed texture, pod color, pod shape, flower position, and plant height. And he counted the plants, thousands and thousands of plants, to make sure he got those ratios. And he came very, very close. And in particular, he found for all these, the traits never mixed, they never blended. There was one dominant, one recessive trait, that the ratio was the same. And so this strongly suggested that what you had here was that for these traits, all the plants of true breeding parents would resemble one of the parent and not the other. That the alternative was a recessive trait. And this is his the basic set of experiments. And from this, we come up with these models of inheritance, that the alleles are discrete. The inherited factor is discrete. We now call them genes. And each individual receives one copy or one allele from each parent. And so for every trait, each individual has 2 alleles or 2 genetic factors. Not all the copies of a gene are identical. Okay, you can have these alternative forms or alleles. If you have 2 that are the same, you're homozygous and true breeding. If you have 2 that are different, you're heterozygous and probably have a hidden trait. Okay, and so these are really important things, and this makes all sorts of sense of patterns of inheritance. Okay, then the alleles never blend, they remain discrete, and the presence of an allele doesn't guarantee expression. Okay, that dominant alleles are expressed, recessive alleles are hidden. Genotypes then can be different from phenotypes. Okay. This is particularly important in later generations, like the F2, which would be the when you're self-fertilizing the results of a cross. Although the hidden traits that were present when you did these crosses can now reappear, and that's when you get that 3-to-1 ratio. That 3-to-1 ratio is really that's the ratio of phenotypes, but if you really look at it, it's a 1-to-1 ratio, where you have 1 homozygous dominant, 1 or 2 heterozygous, and 1 homozygous recessive. So that's what that ratio looks like. And so this is really important. And Mendel expressed this by using, or Mendel didn't, but later on people expressed this by using something called a Punnett Square. Okay, and Punnett squares, where you show the parents, you make a square, and then you show the parents the alleles that the parents probably have on the side of the squares to figure out what are the proportions of the offspring. So on top there you have a white parent, and it's true breeding, so it has 2 recessive copies of the gene here, which are shown as small p's. The purple, true breeding purple parent, has 2 copies of the dominant allele, which is for purple flower color, so it's shown as a large P. And then you carry those across and down to show the possible genotypes. And so in that F1 generation cross, all the plants have one dominant gene and one recessive gene, okay, and they all have the same phenotype. When you allow those to self-fertilize, okay, those F1s each have one copy of a dominant gene and one copy of a recessive gene. And so then you're going to carry those down and then across, and it shows that 3:1 ratio, 3 purple flowers to 1 white flower, and it also shows the genotype ratio, 1 dominant purple, one dominant purple homozygous, 2 heterozygotes, and 1 recessive homozygous. Okay, and so that's a Punnett Square, and we can figure that out. And this is a huge advance, and it allowed Mendel to make inferences that we could then use in domestic animals and could generalize onto people.